Welcome to Cell Biology. Today, we are going to explore one of the most fascinating and fundamental topics in all of biology, mitosis and meiosis. Every single living thing, including you, relies on these processes to grow, heal, and reproduce. By the end of this lecture, you will not only understand how cells divide, but you will also learn how to mathematically track the genetic material inside them. Let us dive right in. To start our journey, let us think about cell division using a simple analogy. Imagine you have a highly detailed instruction manual for building a complex spaceship. If you want to build a second, identical spaceship, you need an exact, word-for-word copy of that manual. This exact copying process is like mitosis, where a cell makes a perfect clone of itself. Now imagine you and a friend want to write a brand new manual together, taking half the chapters from your book and half from theirs. This mixing and halving process is like meiosis, which is used exclusively to create unique sperm and egg cells. Both are just different ways a cell divides its instruction manuals, which we call chromosomes. Now that we have an intuitive understanding, let us look at the formal definition of mitosis. Mitosis is a type of cellular division that results in two daughter cells, each having the exact same number and kind of chromosomes as the parent nucleus. We call this an equational division because the genetic material is perfectly preserved and equally distributed. In this process, the cell undergoes one round of DNA replication followed by a single round of division. This ensures that the diploid state is maintained. This is the typical process for ordinary tissue growth, ensuring that every new cell is a perfect genetic match to the original. While mitosis preserves genetic information, meiosis does something entirely different. Meiosis is a specialized form of cell division that deliberately reduces the chromosome number by half. Instead of two identical cells, it creates four haploid daughter cells that are genetically distinct from the parent cell. Here, one round of DNA replication is followed by two consecutive rounds of division, known as meiosis 1 and meiosis 2. This reduction is absolutely critical. Why? Because when two gametes fuse during fertilization, the normal diploid number of chromosomes must be perfectly restored. If meiosis did not halve the genetic material first, the chromosome count would double with every generation. Understanding these definitions leads us to ask, why are both processes so essential in the real world? Without mitosis, a single fertilized egg could never grow into a fully formed human being with trillions of specialized cells. Every time you scrape your knee, mitosis is the biological engine that generates new skin cells to heal the wound. Meiosis is equally crucial, but for the survival of the entire species. It introduces massive genetic variation through processes like crossing over and independent assortment. If meiosis did not exist, sexual reproduction would be impossible, and a species would lack the genetic diversity needed to survive changing environments or new diseases. To truly master these biological concepts, we must track the genetic material using simple mathematics. We use the variable n to represent the number of unique chromosome sets, and x to represent the baseline amount of DNA. During the S phase of the cell cycle, the DNA replicates. This gives us our tracking rule. The DNA content after the S phase equals 2 times the initial DNA content. However, because the replicated copies remain attached at a central point called the centromere, the actual number of chromosomes does not change. If the initial DNA increases, the replicated DNA increases proportionally. This mathematical approach removes the confusion of trying to memorize phases, letting us logically calculate the outcome of any cell division. Let us apply this tracking rule to a concrete example: human mitosis. We're given a human skin cell with a diploid chromosome number of 46 and an initial DNA content of 2x. We need to calculate the values immediately after the S phase, which is when DNA replication occurs. As we just learned, the DNA is replicated, but the two copies remain attached at the centromere. Because we count chromosomes by counting centromeres, the chromosome number remains exactly 46. However, the DNA content has doubled from 2x to 4x. The cell is now fully prepared to divide without losing any genetic information. This temporary doubling is an absolute requirement before division. Following the S phase, let us see what happens when the complete process of mitosis has finished. The cell enters mitosis with 46 chromosomes and a doubled DNA content of 4x. When the cell finally divides, the attached sister chromatids are pulled apart into two new cells. Because they physically split, each new daughter cell receives exactly 46 individual chromosomes, restoring the original count. Furthermore, the DNA content is halved back to normal, returning to the baseline value of 2x per cell. This perfectly demonstrates the physical meaning of our final answer. Mitosis creates an exact, identical clone of the original parent cell, ready to perform its function in the body. Now that we have tracked mitosis, let us look at the mathematics of meiosis and genetic diversity. The formula we use is C subscript gametes equals 2 to the power of n. Conceptually, this formula calculates the total number of unique genetic combinations possible in the sperm or egg cells produced by a single individual. Let us break down the symbols. The base number 2 represents the two possible homologous chromosomes, one from the mother and one from the father, that can be sorted into a gamete. The exponent n represents the haploid number of chromosomes, which is the number of unique chromosome pairs. As the value of n increases, the number of combinations grows exponentially. Let us put this diversity formula into practice by calculating the genetic variation in fruit flies. We're given that a fruit fly has a diploid chromosome number of 8. We want to calculate the number of unique gametes this fly can produce through independent assortment. First, we need to find the haploid number, n. Since the diploid number is 8, we simply divide by 2 to find that n equals 4. This means there are 4 distinct sets of chromosomes. Next, we substitute n equals 4 into our formula, meaning we multiply the number 2 by itself 4 times. The final answer is 16, meaning a single fruit fly can produce 16 genetically distinct gametes. Having seen how this works in a simple organism, we can now mathematically prove why genetic diversity is so vast in complex organisms. We will set up our proof by applying this same math to human beings. We know that a human has a diploid chromosome number of 46. To use our independent assortment formula, our first step is to determine the haploid number of unique chromosome pairs. By dividing 46 by 2, we establish that n equals 23. This means during the first division of meiosis, 23 pairs of chromosomes will line up and be randomly sorted. This setup is the physical basis for the massive mathematical combinations we are about to calculate. Continuing with our proof, we now take our human haploid number of 23 and plug it into the genetic diversity formula. We calculate 2 to the power of 23. When we compute this exponential function, the result is staggering. Over 8,000,000, 388,000 combinations. This final result mathematically proves our initial statement. Even an organism with a relatively small number of chromosomes generates massive genetic variety for its offspring, purely through the random shuffling of independent assortment. This exponential growth is exactly why no two siblings are exactly alike, and why sexual reproduction is so powerful. To visually reinforce everything we have learned and proven, let us look at this side-by-side comparison diagram. On the left, you can see the conservative process of mitosis. Notice how the parent cell starts with a diploid number, labeled 2N, and ends with two identical diploid daughter cells. The chromosomes simply duplicate and split. Now, look at the right side, showing meiosis. Here, the parent cell undergoes two distinct divisions. In prophase 1, you can see the chromosomes crossing over, mixing their colors. By the end of the process, at the bottom right, we have four distinct haploid cells, labeled N. This diagram perfectly captures the physical difference between cloning and creating genetic diversity. We have covered a lot of ground today, so let us summarize our key takeaways. First, mitosis and meiosis serve completely different biological purposes. Mitosis is the conservative process of cloning cells for growth and repair, ensuring a perfect genetic match. Second, meiosis is the creative process of generating sex cells, deliberately halving and shuffling genetic material to ensure the next generation is diverse. Finally, by tracking variables like chromosome count and DNA mass, we can mathematically predict the outcomes of these divisions. You now have a solid, logical understanding of how life sustains itself and evolves over time.